Alright, students, we're going to be looking at section 4 from your book. It's book C, chapter 3, section 4. It's called the DNA connection, where we're looking at how the code, the DNA code, actually works, how it makes proteins, and how it can be changed. So we'll start with the genetic code section. And it's important to remember that the main thing that genes do is control the production of proteins. So we got to remember that one gene is made to make only one protein. That's a really important fundamental idea. Get my end drawn a little better there. Now, genes are composed of many different chemicals that we represent with letters. The A is a chemical called adenine. The T is a chemical called thymine. The G is called guanine. And the C is cytosine. These all end in -ine because they're all amines. Now, as we move on, we need to understand that these guys will always pair together, and G's and C's will always pair together. You'll never have a G paired to a T. It just strictly does not work that way. Now, we're not going to use the chemical names too much anymore. We're going to stick to the A's, T's, G's, and C's, just like you see here. So, as we're moving on, we're going to talk about the order of these things. The order is very important. It's what actually tells what material is going to be made by a certain gene. The order of nitrogen bases, and those little letters are nitrogen bases, determines the genetic code. So, if I have A, C, T, G, C, C, A, C, T, G, A, C, which looks like it's long, but it's actually a very short string of letters, that could code for a couple amino acids. The genetic code, has 4 letters, A, C's, G's, and T's, and each word in the code is 3 letters long. We call the 3-letter word a codon. So there's a word, there's a word, there's a word. So this string here can code for 1, 2, 3, 4 separate amino acids. So this could make a protein that's 4 amino acids long. Okay? Now, 2 of these, here and here, will be the same amino acid because they have the same word, the same codon. Okay, and these can be anywhere from a couple hundred letters long to 1,000,000 or more letters, depending on how big and complicated the protein needs to be. So remember, the order of the bases forms a code that tells what protein will be produced. Now, we'll look at how cells make proteins. Now, during the protein synthesis, the cell uses information to produce a specific protein. This happens in the ribosome. So remember that we keep our DNA in the nucleus for a reason, because there it is very safe, and we do not want to take our master recipe copies out of the nucleus because they could be damaged in the cytoplasm. So we always make a copy of the DNA, and that's called a messenger RNA. Now, RNA is very much like DNA. We'll have our DNA here, and that would be like T's, C's, A's, G's. Of course, the binding is a little different, T's to A's, and so on. But your RNA will have the same codes, but no T. And in place of a T, there will always be a U if you're an RNA. So if you're looking at a strand and you see any U's, you know that it's an RNA strand. If you see any T's, you know that it's a DNA strand. And that's how you're going to tell the difference. So we start with our strand. Let's say that we got a TTAC or GCAGCTACGATC. We'll stop there, that's plenty. To make this into an RNA, we remember that T's bond to A's, so we're going to draw our RNA strand up here, and we're going to say T bonds to A, T bonds to A bonds to U, because remember in RNA, there is no T. G bonds to C, C bonds to G, A to T, I already forgot the important rules. We better fix that. A bonds to U, G bonds to C, C bonds to G, T bonds to A, but A bonds to U, not T. Another G, a C, a U, an A, and a G. So this gives us our RNA strand. This guy can be carried out of the nucleus. So we're going to take him out of the nucleus, which means we're going to leave the rest of this stuff out right here for now. So we're just going to move him away, except for a few parts that we forgot here. So now we just have our RNA strand. This is pretty important stuff right here. We've got our strand. Let me go ahead and clear up our image real quick here. We're going to take this RNA strand, and it's going to have a ribosome lock onto it. Now the ribosome is kind of a funny looking thing. It has space for 2 codons, or 2 words. So it will lock on to 6 letters. It has a large, what we call a subunit here, and a couple spots for what we call transfer RNAs, or tRNAs, to fit in. Now, as we're looking at this guy, it'll be a little tricky to kind of represent some of what's going on here. I'm going to take some sides off of the face here and patch up the image so we can make this work a little better. This was an A, this was a U, that was a C, I believe. So that'll do. Now here's our ribosome. What's going to happen first is a transfer RNA, let's show those guys in green, is going to appear and lock into this first spot. Now every transfer RNA is different. It'll have kind of a funny shape. I draw them like this, but how they actually look is the subject of a little bit of debate. And at the bottom here will be 3 letters, 1 word, but it's the anti-word or the anti-codon for 3-letter word on the strand. So we got AAU there. That will be a UUA anticodon. So this anticodon is going to move and lock in right here because his 3 letters, the U, U, and A, match the AA and U, and they attach just like a magnet. And the second spot, I'm going to use a different color so that maybe it'll show up a little easier. We'll use blue. You got CGU, which is going to lock a GCA transfer RNA into this spot. So we got our 3-letter anti-word there, sticks in just like a magnet, and away it goes. Now, as we're moving along, we got our 2 transfer RNAs in there, and these guys are carrying a payload of some amino acids. Let's say this one's carrying a square, and this one's carrying a triangle. We're not going to worry about the actual names of these amino acids. We can deal with that later. So these guys will transfer this stuff here to the ribosome, and then we're going to slide our ribosome down the strand. This will be a little tricky to represent. I'm going to try to do it, but we're probably going to pick up some extra stuff and move it with us. So just try to use your imagination. So we slide over 1 3-letter word. Now we've got some letters that don't match up. We've got a CGU right here, CGU, but UUA is still the anticodon. So that no longer matches, so this guy is going to be repelled out and away from the ribosome. So we're going to go ahead and get rid of this guy. Getting rid of all his letters will be a little trickier, but I'll see if I can do it. This is almost close enough. I think we can work with that. Now the GCA one, he's going to move just 1 spot to the left because this still matches his code. So we're going to move him over, and he is going to bring with him his amino acid. And this amino acid is going to be added to a growing chain of amino acids over here. So then we tie that amino acid onto the previous one because this GCA transfer RNA carried the triangle and the recipe called for a triangle. Then we're going to add our next codon. A CGA here means it's a GCU, must be in this spot next. So our transfer RNA will appear. Go ahead and draw him in. And let's say that this one is carrying a circle. So this guy has got a circle. Then we slide the whole strand down again. It's going to get a little sloppier, of course, but let's try that one more time. Slide down, one more 3-letter code. Then we take the transfer RNA with the newest amino acid over here, attach it to the string. Of course, this string here and this string here are going to move off, especially if I can get them selected right. The transfer RNAs are moving. The proteins that they're carrying, or the amino acids they're carrying, get added to our protein, and this continues until we reach the last word, or the last codon. The last codon in a gene is a stop codon, and it tells this process to end. The ribosome falls apart, our completed protein gets thrown off, and everything has stopped. Now once this messenger RNA is turned loose, he can have another ribosome attached to it and start this process again. In fact, once the ribosome has moved down the strand a little ways and there's enough room, we can attach 2 or 3 or 4 ribosomes to the same strand and be reproducing proteins several different times from the same strand. So that's the way that we make a protein with a ribosome. We copy the DNA into a messenger RNA. We attach a ribosome to the messenger RNA. It starts reading the 3-letter codes, and transfer RNAs with matching 3-letter anti-codons come carrying specific amino acids and adding them 1 amino acid at a time to the growing chain. That keeps going until each transfer RNA has added what its requirement for the recipe is. You hit a stop sign at the end of the strand, and the process is complete. Now we need to talk about mutations. When we're talking about mutations, you can't be thinking about the X-Men because that is just not how things work. The important thing about mutations is to remember that it is any change in the DNA. Most mutations don't do anything, have no effect on you. There may be a small effect, but what we really consider a mutation to be helpful or harmful is if it makes it easier for an organism to survive and reproduce, or if it makes it harder for the organism to survive and reproduce. Those are the important judges of whether a mutation is good or bad. It has to improve or hurt your ability to survive. If it doesn't affect your ability to survive, it's neutral. Now, a mutation can be a small change. Well, just look at a DNA strand here, AC or AGCT, ACG, ACC. It can be a very small change, such as maybe changing 1 letter from an A into a T. That's a mutation. We could actually completely delete 1 or 2 letters. That's called a deletion. We'll learn more about that later. So we could have this happen and close up the strand. We could have letters added, so where we have a complete gene and then we end up with a couple added or maybe just 1 added. Or we can have strange things like an inversion, where a piece of the strand can actually be removed and get stuck back in upside down. So then we got CGA CGA instead of AGC CGA like we had before. So we can end up with strange things happening to cause these mutations at the letter level, or we can end up with entire pieces of our chromosomes. Remember, our chromosomes, they split and divide. Now as they're splitting, let's say this one is going to move to this side of a cell, this one is going to move this way to this side of the cell. Sometimes these guys can get stuck together Or perhaps maybe just part of this guy might get stuck to the other chromosome. So his